So I'm going to share with you today an exciting project that has been going in the lab over the several few years, and really is the heart of the proposal, the research plan that I submitted to the Beutler Foundation. And it's essentially a technological advance, a new method to detect cell death, which we think has extensive medical implications. Papers describing this method are starting to come out. How do I move here? Okay, so the general field that I'm going to talk about is this emerging new type of biomarker, very exciting, so-called liquid biopsies, the concept of using circulating cell-free DNA to diagnose and detect diseases. Unlike circulating tumor cells, these are circulating fragments of DNA. And we've known for decades that when cells die in the body, in many cases, they release small nucleosome-sized fragments of DNA that go to the circulation. We don't know much about the process of release of those fragments, but we know that they come from dying cells. We know they have very short half-life. They appear to be cleared by the liver within 15 minutes to 2 hours. So essentially, they represent cell death events that took place in the last hour or two. And we know that the numbers, there aren't many such fragments. There are about 1,000 broken genomes, so genome equivalents per milliliter of healthy plasma. In various pathologies, this goes up. And really, after knowing that for over 40, 50 years, in the last few years, there's really a boom of using this phenomenon with the force of next-generation sequencing for diagnosis of pathologies. So what's already commercially available is really transformed prenatal testing, is so-called NIPT, non-invasive prenatal testing. The idea is that if you about 5% of the DNA circulating in maternal plasma early in gestation is actually coming from placental cells of the fetus that were shed, died, and released their DNA. So if you sequence maternal plasma from her systemic circulation, you can identify essentially the genotype of the embryo, and this is used now to detect Down syndrome in cases where there's, higher than expected proportion, for example, of fragments coming from chromosome 21. What's coming soon is really a very bubbling scene in the academia and industries in the context of cancer. The idea is that one can use such liquid biopsies to identify somatic mutations of the tumor. What's being done already, I think, is using that as a way of monitoring disease tumor processes in the tumor, like progression, response to therapy, essentially genotyping the tumor without touching the tumor. And the fantasy is that this can one day turn into a way of early diagnosis, but we're not there yet. Another smaller field concerns the detection of rejection of transplanted organ based on SNPs, single nucleotide polymorphisms that distinguish between the donor tissue and the recipient. When you find such fragments of circulating DNA that carry those SNPs, that's an indication for excessive cell death in the transplanted tissue, be it liver, kidney, heart, and others. So that's a very busy field already, but we have come to that from different perspectives. So in all these three applications, the principle is that you detect a tissue that has a different genome than the host, right? The fetus, the tumor, or the transplanted organ. What do you do in cases where there's cell death in a tissue that has a genome that is identical to the host, like the vast majority of cell death events in our body? And put the question another way, another direction is how can we identify the tissue origins of circulating DNA that contains a normal genome? And this is, so this is the method that we came up with. This is probably the most important slide of the talk. So we know, of course, that's very basic, right? Every cell type has a unique, has the identical primary DNA sequence, but every cell type has, by definition, a unique DNA methylation signature, which is really part of, this defines to a large extent what is cell type identity. DNA methylation, we know a lot, again, from the studies of Howard Sitter, that it's a major method of controlling gene expression, essentially cell identity. And we know a lot about that. We know that methylation patterns are really, on DNA, are really relatively stable in health and disease conditions, and they're conserved among cells of the same type, and also among cells of the same type in different individuals. And I'll show you a few examples that we actually use in our work. So, oops. So, for example, the insulin gene is expressed only in pancreatic beta cells, and studies by Wally Gilbert, I think almost 30 years ago, showed that the insulin gene promoter is unmethylated only in pancreatic beta cells, the only cell type that expresses insulin, and the insulin gene promoter is heavily methylated elsewhere in the body. So this is the heart of the idea, right? If you find there are about 1,000 fragments of the insulin gene promoter circulating in your plasma, but most of them will be methylated because they come from all kinds of tissues, but every single molecule of the insulin gene promoter circulating in plasma that is unmethylated can come from only one place, right? And must represent a single beta cell that has died in the last hour or so. So that's the heart of the method. And this goes way beyond that. The surfactant gene promoter we found that, is unmethylated only in lung epithelial cells and methylated elsewhere. Albumin gene promoter is unmethylated in hepatocytes, allows expression of albumin and methylated elsewhere. So this is the basic concept. By identifying the methylation status of marker loci, we can infer cell death in specific tissues. So the flow chart, I won't go into much detail, but it starts from a very extensive bioinformatic pipeline that we had in the background. I'll go into that in one more slide in a bit more detail. Really, we built over the last few years a big atlas, a human methylome atlas, where we have essentially all human cell types or tissues and their methylomes. And by comparing the methylation status of different sites in the genome, we can identify many loci that are unmethylated specifically in one cell type and not in all other cell types. These will be our markers. And then the procedure is really to collect blood, isolate plasma, isolate circulating DNA from that, treat with bisulfite, which is a classical method of chemical that is used to distinguish between methylated and unmethylated DNA. I won't get into that. But the final stage is really to PCR amplify the sequence of interest, the marker locus, and then sequence that and essentially determine the fraction of circulating DNA molecules in plasma that have this tissue-specific methylation pattern. And from that, you essentially can infer how many DNA molecules are circulating that come from tissue X, and we interpret that as an indication for the rate of cell death in that tissue right now. I mean, close to the time of sampling. And from now on, I'll show you a few examples of utility. This what turned out to be a very broad and generally useful method. Just one more thing about this atlas. So this is a short fragment or a small piece of our atlas. So the columns are multiple human tissues or cell types, and the rows are sites in the genome that can be methylated or not, and the color reflects the methylation status. And when we align, this is just a small fragment, a small fraction. We show here only the top 20,000 sites. The actual matrix has 500,000 sites, and we have hundreds of columns of tissues or cell types. So look, just one example. So here is the beta cell methylome. This is one. Much of it comes from public sources, but some we've done in the lab. Nobody bothered to isolate human beta cells and prepare their methylomes. So when you align it this way, you see here clearly a cluster of sites that blue is unmethylated. They are unmethylated in beta cells, like the insulin promoter that I showed before, but many others, and are fully methylated elsewhere. So these will be excellent markers to tell apart DNA coming from beta cells from other cell sources. And we find that easily in every tissue that we find, and not surprisingly so, because this is really what distinguishes cell type from cell type. Most of what my lab has been doing is really about diabetes and pancreatic beta cell biology. So this is where we started. This turned out to be the most difficult task, but this kind of leads the technological development. So just to show you the principle of the method. So here is the insulin gene, structure of the insulin gene promoter. There are several cytosines. And what you see at the top is the methylation status of each of those individual cytosines in different tissues. Right? So cytosine number 1 over here is, as expected, fully unmethylated. This is the degree of lack of methylation. Unmethylated in beta cells, and is methylated or there are almost no unmethylated molecules in DNA coming from all other tissues. Most importantly is blood, which is the main contributor to cell-free DNA and therefore the contaminator. Cytosine number 2 is also showing the same 3, 4, 5, 6. So this is good. There's a signal, but it's not a good enough signal to noise ratio because this will not allow you to detect the DNA from a beta cell that is embedded among 1,000 or 100 molecules from blood, for example. But the trick was the realization that actually DNA methylation comes in what we call now haplotype blocks. So if you look at you now count only the molecules of the insulin gene promoter that are fully unmethylated in all 6 cytosines. This is the rightmost columns. In beta cells, the vast majority of molecules are really fully unmethylated. But in non-beta cells, you never ever see that. So each of those small bars is really is an accident, right? But never ever will a blood cell have all 6 cytosines in the insulin gene promoter be unmethylated. This happens only in beta cells. So when you look at the clusters of multiple cytosines, you get a dramatic improvement of signal to noise. So now we're ready to go to blood and to ask what's in the plasma. So the plasma of healthy individuals has no whatsoever cell-free DNA coming from beta cells. Not surprising. That's a very static tissue. It doesn't die normally in a healthy individual. Perhaps the most dramatic indication came from a collaboration with James Shapiro, who is the transplantation surgeon at University of Alberta that established islet transplantation. So these are plasma samples from people with long-time type 1 diabetes. They have no beta cells anymore. They've been transplanted with cadaveric islets from brain-dead donors. And James is collecting plasma samples for us from before, shortly after, 1 day, 1 week, 1 month after transplantation. And what you see here, there's nothing before transplantation. These people have no beta cells that can be killed. But shortly after transplantation, there is a huge peak in each of those patients of beta cell DNA, representing DNA coming from the graft. And we think it actually represents 2 processes. That what we see immediately after transplantation is a process that the transplant surgeons called IBMIR, for instant blood-mediated inflammatory reaction. But perhaps more interesting, we see also a variable degree of cell death of beta cell DNA at later time points, probably representing either an immune attack or toxicity of the immune suppression drugs. And we've done many more of that and recently realized that this actually can help us predict the outcome of the graft. So those that graft recipients that will be independent of insulin 3 months after transplantation, that is a successful graft, can be predicted by not having cell-free DNA 1 day after transplantation. And those that shed lots of beta cell DNA to circulation shortly after transplantation, these are the expected failures. There's much more to do here that we intend. Of course, the most important challenge is to increase the sensitivity of the method to allow for what we don't have today, which is preclinical detection of beta cell death in people at risk for type 1 diabetes. We can't diagnose type 1 diabetes today before the onset of hypoglycemia, which is too late. Is there beta cell death in type 2 diabetes? And what else is dying in diabetes? Other cell types, there's emerging evidence for involvement of the exocrine pancreas. Immune cell dynamics must be dramatic in type 1 diabetes, etc. I want to give 2 other examples. So that's a beautiful, shorter, I think. So that's a beautiful marker of cardiomyocyte DNA, right? We pulled that from that screen. We don't even know which gene this sequence in the DNA is regulating, but look at its behavior. It's fully methylated in blood and all tissues. It's unmethylated in about 50% of the DNA from the heart. And it's almost fully unmethylated in DNA from purified human cardiomyocytes, which makes sense because the heart contains lots of non-cardiomyocyte DNA, so highly specific. That's a routine control experiment that we do. We mix cardiomyocyte DNA with known proportions with blood DNA, and using the assay, we can calculate exactly how much we injected into the mix, and this goes up to being able to detect 1 molecule in 1,000. So the assay is highly specific. Again, what we see in plasma, so healthy individuals have almost no cardiomyocyte DNA in their plasma. Again, not surprising. These are patients from the Hadassah ER, people that showed up with chest pain and were diagnosed as having acute myocardial infarction. They have lots of cardiomyocyte DNA in their plasma, as expected from a dying heart. The kinetics of the increase of cardiomyocyte DNA parallel what we know from the classical, the gold standard clinical marker, which is troponin. So at admission, there are low levels because the dead material is trapped within the dead heart. But then when our interventional cardiologist collaborator is performing angioplasty, the dead stuff is released, and there's a huge spike of both troponin and cardiomyocyte DNA. Troponin goes away slowly because of the low clearance rate. Cell-free DNA goes away quickly. And the assay can actually tell from those coming with chest pain whether they have or don't have myocardial infarction. We then moved on to something that was not a positive control, but more of a real question. So it's known for a while that patients in the intensive care unit, many of them show high levels of troponin, specifically patients with sepsis. In the ICU units show high levels of troponin. And nobody really knows whether this reflects a silent death of cardiomyocytes. They don't show up with clinical signs of cardiac failure, usually, or maybe just a non-lethal leak of cardiomyocytes. They could open up, release some troponin and be sealed back, or maybe reduce clearance via the kidney. So we used the assay to ask whether there's cell-free DNA coming from cardiomyocytes. And surely there is plenty of it. Right? So sepsis patients, that's strong evidence that in sepsis patients, there is actually a strong, a massive process of cardiac cell death. And what was most striking, I think, is that if you score the patients by the amount of cardiac cell-free DNA that you find in plasma, this is a pretty good predictor of who's going to die within 3 months. Which, retrospectively, the anesthesiologist that collaborates with us says, well, if the engine fails, no surprise that the patients will die. But that's a good predictor. We're now moving on to other directions. For example, there are huge levels of troponin found in the blood of marathon runners after a run. These are usually healthy people that don't tend to die, but do they harm their cardiac muscles? So we're about to check. We see also high levels of cardiac DNA in the plasma of newborns. We think this reflects part of the normal process of cardiac development and turnover in the perioperative setting, in people receiving chemotherapy. A lot to do there. So I'll summarize, and then 2 more slides of data. So we think that this method in its general form can be really used as a novel method for early detection of disease. We think about type 1 diabetes, cancer, fatty liver, neurodegenerative disease. We have nice data with detecting neuronal cell-free DNA in patients with ALS, assessing response to drugs, assessing toxicity of drugs, and going back to my original identity as a developmental biologist, I think that's a powerful tool to study human tissue dynamics that were inaccessible so far. For example, we know nothing about adipocyte turnover in humans, but we have now beautiful markers for adipocytes, so we can assess whether adipocytes die, not just shrink and expand, as happens in diet, what happens in early postnatal life, what is aging. But the vision really is to move from what we call directed markers, as I showed you, to something that will be more general, really a novel blood test, general one, to spot abnormalities. And the imagination is something like what used to be in the first generation of Star Trek, right? So a machine that will assess the levels of damage to different organs and deviation from the norm based on that blood test. And the truth is that we've started to do that, and it seems to be working pretty well. I'm finishing with this. So the concept here is to obtain the complete methylome of plasma of an individual, compare that to the same matrix from before, showing the methylation pattern of different cell types, and deconvolute that signal, which is essentially an equation, right? Solve the equation and figure out which cells have contributed in which proportion to that mix that you see in plasma. And it seems to work. So this is my own plasma, methylome, deconvoluted. So in my own plasma, 30% of the cell-free DNA comes from dead granulocytes, neutrophils, monocytes, lymphocytes. a large proportion from erythrocyte progenitors. Note that unlike all other cases which reflect cell death, this actually reflects cell birth, right? When erythrocyte progenitors shed their nucleus, that's what you see here. This reflects the birth of red blood cells, enucleated. And perhaps most important are the contributions from solid tissues. So about 10% of cell-free DNA comes from endothelial cells, and 1% from hepatocytes. So relatively quiescent tissues, but so large that even the baseline turnover gives you that. As you know, initial evidence that we know what we're talking about, so this is again me, but this is samples from a patient that was transplanted with islets an hour ago, right? So a huge increase in the total levels of cell-free DNA. The algorithm automatically identifies DNA coming from the graft, the beta cells and cells from the exocrine pancreas, essentially what was transplanted. And what came as a surprise to us and also to Dr. Shapiro, much of the contribution actually reflects a very robust immune response to the foreign transplant that was put in. So that's interesting. And this moves now in many other directions, this concept of deconvolution, just one last piece of evidence or something that makes us very puzzled. So along that process over the last few years, we've collected thousands of plasma samples from healthy individuals, and for most of them, we prepare cell-free DNA. We measure initially just how much there is there before even analyzing. And we see an interesting U-shape, the U-shape of life, right? So newborns have a lot of cell-free DNA. This goes down with age. Your best years are 30 to 40, and then you start to climb until very old age. These are people that we sampled from nursing homes. We are very curious to understand what's underlying this. We think in the children, that's probably the process of development, turnover associated with development. This might reflect some decay associated with aging. So I'll stop here. These are members of the group. This is a very close collaboration with Ruti Shemer, a long-time methylation expert, and Ben Glazer, who is my close partner, collaborator, is an endocrinologist at Hadassah. Tommy Kaplan is a computational biologist. Much of it is really after conversation and discussions with Chaim Sider here, and very important collaboration with multiple clinicians that share their knowledge and samples. Thank you.